This discussion will continue our look at the innate immune response. We're going to take a look at the second line defenses. Second line defenses are usually seen internally, and they consist of various enzymes and proteins and pathways that will function internally to try and prevent infection or invasion of pathogens. These second line defenses, although they're not just simply skin and mucous membranes, are still going to lack specificity, and also they will lack memory. So we'll begin by taking a look at the top line of this chart. And you see they say as a generic category, we're going to evaluate here just for a moment or two, antimicrobial substances. It's kind of a broad term that just refers to various proteins and other components found within the internal environment that are going to have antimicrobial properties. So we begin here with interferons, abbreviated IFNs. Interferons literally interfere with the ability of a virus to do what we call replicate. We do not consider viruses to be living organisms due to the fact that they lack the ability to reproduce on their own. Due to this inability to reproduce on their own, viruses have to essentially invade other cells and hijack or take over that cell and cause that cell to produce copies of the virus. The process of making copies of that virus we refer to as viral replication. And in order to accomplish viral replication, essentially what the virus will do is introduce its own genetic information, its own DNA or its own RNA, into the host cell, and the host cell begins to copy that DNA or RNA from the virus instead of its own, and ultimately the cell will begin to make new viruses instead of rather healthy proteins. Well, as it somewhat implies, interferons interfere with the ability of a virus to accomplish replication. It prevents that virus from being able to interject or introduce its genetic information into a new host cell. So, as you see here, the interferons have a little bit more narrow scope. It's not effective against all pathogens. You wouldn't see interferons be effective against bacteria, for instance. But there is still a lack of specificity from the standpoint that these interferons are effective against a broad variety of different viruses, not just one specific virus. So that allows them to be lumped here up underneath the innate immune response. Next, we see the complement system. And we'll talk more about this complement system later. But for right now, I just want to introduce the concept to you. The complement system is a series of 3 complementary reactions, hence the name complement system. The 3 reactions, and you will need to know all 3 of these reactions that occur during the complement system, are going to be phagocytosis, inflammation, and also cytolysis. So the complement system, once activated, leads to those 3 reactions, and ultimately it is those 3 reactions that help accomplish the immune response. Once again, that's phagocytosis, inflammation, and also cytolysis. Next, we have iron-binding proteins. Iron-binding proteins do exactly what their name implies. They bind iron. If you look at many bacteria, several of their enzymes will need or require iron to function as a coenzyme in order for the enzyme to work. So ultimately what happens is by binding up the iron, you can prevent certain bacterial enzymes from working, and this may slow down their growth, slow down their ability to divide and reproduce. If there are fewer bacteria, then you have a greater likelihood of being able to eliminate the bacteria that are there. So iron-binding proteins sequester or bind iron, essentially limiting the iron available to the bacteria and therefore slowing down their growth. And then finally here we have antimicrobial proteins, a broad lumped together category that your book uses for various proteins that have antimicrobial activities. These antimicrobial activities could include anything from enhancing phagocytosis to initiating inflammation. And there's a variety of different proteins, so there's nothing specific to try and keep in mind, but like I said, they have antimicrobial functions as far as once these proteins are seen, they can lead to inflammation or enhanced phagocytosis. Next, we have natural killer cells. Natural killer cells are actually lymphocytes. However, these natural killer cells, or NK cells for short, lack one property that the T lymphocytes and the B lymphocytes have, and that's going to be specificity. Now, as far as how these natural killer cells work, they work pretty much the same as your CD8 cytotoxic T cells that we'll talk about later in this chapter. So I'll tell you, or remind you at least when we talk about those CD8 T cells, that the natural killer cells work in the same manner. But for right now, these natural killer cells are cell killers essentially. They go out and destroy other cells, other human cells that essentially should not be present in the body. These targets could include cells infected by a virus, cancerous or tumor cells, and potentially also tissue transplanted cells, because technically speaking, that tissue that has been transplanted into another person's body, it really shouldn't be there. It's not of their body, so your immune system should try to destroy it. So, like I said, these natural killer cells are cell killers. They function basically the same as CD8 cytotoxic T cells. The one key difference between natural killer cells and CD8 cytotoxic T cells will be the natural killer cells lack specificity. Alright, next we want to talk about the phagocytes. The phagocytes accomplish phagocytosis, which just means that they ingest, as it says here, foreign or particulate matter. Now, I do want you to recall that there are three types of phagocytes. We mentioned these white blood cells when we talked about blood in chapter 19, I believe. The three phagocytes that you should keep in mind are neutrophils, macrophages, which ultimately become monocytes once they're active, and also eosinophils. So don't forget about those three cell types that are phagocytes. These cells, as it says here, accomplish phagocytosis, and we need to take a quick look here at the 5 phases of phagocytosis. We'll begin over here at the far left-hand side with the first bullet, which is chemotaxis. This is a chemical attraction. Tissue that is infected or damaged may begin to release what are called cytokines. Cytokines are basically chemical messengers, and they're how cells communicate with one another. These chemical messages, these cytokines that get released by the damaged tissue, can then begin to attract phagocytes to the damaged area. Next we'll see adherence. Adherence is the process of that phagocyte allowing the target microbe or target matter to stick or adhere to its cell membrane. Usually this is a function of membrane proteins found in the membrane of that phagocyte. Next we have ingestion. As it would imply, ingestion is bringing the target microbe into the phagocyte. This happens as pseudopods, which are extensions of the cell membrane, extend out and begin to surround the target microbe. As they do, once the extensions, the pseudopods, come together, they would form a small tiny vesicle called a phagosome. And at this point, the microbe or target matter would be ingested. These phagocytes contain digestive organelles called lysosomes. These digestive enzymes found within lysosomes are going to be allowed to attack the target microbe as the phagosome and lysosome fuse together to form what's called a phagolysosome. As the enzymes mix with the microbe, they will begin to attack it, and we transition now into the fourth phase of phagocytosis, which is digestion. The enzymes from the lysosome are allowed to do their job, which is to break down the microbe. Finally, as that digestion continues, we would ultimately end up killing that targeted microbe, and then sometimes you have indigestible material remain behind in what's referred to as a residual body. So keep in mind the 5 phases of phagocytosis. Be able to put them in their proper order. And you should also, at least from name, be able to interpret or surmise what is going on a little bit in each one of those processes. So we once again had chemotaxis, adherence, ingestion, digestion, and then killing. So we go back here to the chart. We'll next see or talk a little bit here about inflammation. Inflammation is a non-specific innate immune response. It has 3 main goals. Those goals are confinement, destruction or killing, and tissue repair. So the goals of inflammation, once again, are to confine the potential infection, destroy the microbe responsible for that infection, and then to allow the tissue to begin to repair. Now you absolutely have to remember what is at the core underlying cause of inflammation. That comes down to one simple process. That process is vasodilation. At the core of it all, inflammation that is, vasodilation is what causes it. So let's take a look at a diagram and talk a little bit more here about inflammation. As you see here at the top of the diagram, there's some type of injury to this tissue. This has allowed microbes to enter an area of the skin where they usually are not found. Technically speaking, this would be an infection. Due to various cytokines that are released, we get vasodilation, and this would lead to inflammation. So how is inflammation going to help or lead to the confinement, destruction, and repair of this tissue? Well, vasodilation causes the endothelial cells lining the blood vessel to become a little bit more spaced apart. And essentially, because of that, the vessel becomes leakier, or we say it has increased permeability. Due to the increased permeability during the process of bulk flow, more fluid will begin to escape out into the surrounding tissues around this vessel. The extra added fluid will ultimately cause an increase in pressure. And this increase in pressure will make it very difficult for this microbe to begin to move, essentially confining it to this area where it is now. So the increased fluid, the increased filtration, the increased interstitial fluid that results from the leakiness of the vessels helps in the confinement. Let's next talk about how this vasodilation can lead to an increased destruction of the target microbe. What comes along with the now dilated vessel will be more blood flow to the area. More blood flow means more white blood cells coming. So these additional white blood cells can help in the destruction of the target microbe. Not only do you get additional blood cells, however, but remember these white blood cells have to do something called immigrate from blood into the surrounding tissues. Due to the increased permeability of the vessel, these white blood cells now have an easier time immigrating. So we get more white blood cells and easier immigration. That helps or aids in destruction of the target microbe. Next, let's talk about tissue repair. For tissue repair, it's quite simple. You need nutrients in order to repair that damaged area. The only way you get additional nutrients to the area is by increasing blood flow. So since the vessel is dilated, you get more blood, therefore you get more nutrients, and the extra nutrients help or aid in tissue repair. So that's how vasodilation, which is the root cause of inflammation, can aid in confinement, destruction, and tissue repair. Now, for the purposes of your quiz and exam, you also need to keep in mind something that we refer to as the 4 cardinal signs and symptoms of inflammation. And they're honestly all linked back to vasodilation. The first of these cardinal signs and symptoms is redness. The area gets red due to the increased blood flow. Now, if we're dealing with the skin, you may see an alternate term for redness, which is erythema. That's Erythema. It's just a reddening of the skin. So redness or erythema, first characteristic here of inflammation. Secondly, we get swelling or edema. The swelling or edema results from the additional filtration of fluid into the surrounding tissues. So that extra fluid causes swelling. Thirdly, we have pain. The pain results usually from cytokines that are released and that affect nearby nerves. So pain. Fourth and finally here, we have heat. Heat is a result of the additional blood flowing to the area. The additional blood flow brings more blood, obviously, but remember, blood is a warm fluid. So the additional blood flow not only brings more blood, but it also brings more heat to the area, therefore the area heats up. So, redness, pain, swelling, heat. 4 cardinal signs and symptoms there of inflammation. Please keep those in mind. Finally, to finish this second line defense discussion out, we have fever. What I always have to emphasize to everyone is that fever and heat are not the same thing. Heat is a localized increase in temperature due to the vasodilation that occurs with inflammation. Fever is a general resetting of your entire core body temperature. So your entire core body temperature goes up. Cytokines, usually referred to as pyretics, affect your hypothalamus, and these pyretics cause the hypothalamus to basically reset the thermometer, increase it, raise it up a little bit. But that increase in body temperature does come with some benefits. The first of those is that it can intensify the effects of some antimicrobial substances. What happens is when the core body temperature is increased, the cell membranes become a little bit leakier. And those leakier membranes will allow antimicrobial substances to penetrate cells that may be infected a little bit easier. This will help improve or intensify the effects of those antimicrobial substances. Secondly, the increase in temperature may inhibit the growth of some organisms. Bacteria, for instance, like many organisms, like to grow over a specific range of temperatures. Well, if the temperature range is not adequate or not optimal, then their growth may be inhibited. Once again, that may give the slowing of growth may give your immune system a chance to catch up and get a hold of that particular infection. And finally, if you think back to chemistry, increased temperature just simply speeds up any reactions that are occurring within the human body. So a slight increase in temperature may mean that all the reactions necessary to help eliminate the various pathogens or help in the repair of tissues will all be sped up, therefore allowing healing to occur quicker. So fever is an increase in core body temperature. As benefits of fever, you get increased efficiency of antimicrobial substances, inhibiting the growth of some microorganisms, and also an increase in the rate of reactions that are going to allow for healing and repair. That's the last of the second line defenses, so we'll stop there with this discussion.